Welcome to the Deep Dive, the show where we take the source material you share and synthesize the most critical insights into instant understanding. Today, we are diving deep into a really fundamental question in biology, especially when you think about cancer or how we develop. It's this: how does a cell decide what to be?
It's the ultimate cellular question, right? Nature versus nurture.
Exactly.
When you look at a single cell inside say, a complex tumor, how much of what it's doing, its identity, is because of its ancestry? Its family history, what it inherited.
And how much is because of its environment? The neighborhood it lives in, the signals it's getting from all the cells around it.
Right. Is it intrinsic or extrinsic? That's the puzzle.
And for a long time, trying to answer that has been, well, it's been a huge technical challenge. We've had these amazing tools for tracking a cell's family tree, its lineage, things like clone tracing, which use these random DNA barcodes.
And on the other hand, we've had these incredible imaging methods like spatial transcriptomics that can show you exactly where every gene is being turned on inside a tissue down to a single cell.
But the two just didn't mix.
Not at all. They were completely incompatible. The high-res imaging platforms like Ceph, they're designed to look for known sequences, actual genes from the mouse or human genome.
They need to know what they're looking for at a time.
Precisely. So they had no way of seeing the totally random DNA sequences that the lineage tracing tools were using. It meant you had to make a choice. You could either know the family history or you could know the location. You couldn't have both.
So the mission was pretty clear. We needed some kind of bridge, a translator that could get these two powerful technologies talking to each other.
And that's the breakthrough we're looking at today. It's a system called, fittingly, SpaceBar.
SpaceBar. I like it.
It's this custom-engineered barcoding strategy that for the first time lets you do single-cell clone tracing at the same time as you're mapping out all the gene expression in the tissue.
Using the same standard lab workflows people already have.
Exactly. And the whole point is to finally really be able to tease apart what's coming from the cell's internal history versus what's being dictated by its external environment.
Okay, so let's unpack that. The key seems to be moving away from random barcodes to something custom designed. How did they actually engineer 96 new barcodes that can fool these really sophisticated imaging systems?
Well, that's the core of the engineering. They essentially built them from the ground up. They're synthetic, which means they don't exist anywhere in the mouse or human genome. They're totally unique or orthogonal, as scientists would say.
So there's no chance of the imaging system getting confused and seeing a real gene instead.
No chance at all. And they made sure these sequences were, robust. They're about 1,000 base pairs long, which gives plenty of room for the imaging probes to lash on, making the signal really strong and clear. They basically look and act just like a normal piece of RNA.
But wait a second, 96? That doesn't sound like enough to track, say, thousands of different cell families in a tumor.
But that's the beautiful part. It's not about one barcode, one family. The real power comes from something called combinatorial labeling.
Okay, break that down for us.
Think of it like a lottery. They package all 96 of these unique barcodes into a library of viruses. Then they infect the cells they want to study at a really high rate, what's called a high multiplicity of infection, or MOI.
What does that mean for the cell on the receiving end?
It means instead of just getting one lottery ball, one ID tag, each cell gets a whole handful. It gets a unique combination of those 96 possible tags.
I see. So my cell might get barcodes 5, 23, and 81, while your cell gets 12, 50, and 92.
Exactly. And by changing how many viral particles you give each cell, you can tune the number of possible combinations. It skyrockets from just 96 options to hundreds of thousands, maybe even millions of unique clonal IDs. Any two cells that have the exact same set of barcodes must have come from the same original parent cell. They're sisters.
Got it. The pattern is the signature, not the single tag. So before they took this into a complex tumor, they must have tested it in a simpler setup, right? Like on a petri dish.
Of course. They started in vitro with melanoma cells. They profiled tens of thousands of them. And the detection was fantastic. Something like 89% of the cells had at least one barcode.
And what was the key finding there? Even on a simple plastic dish where the environment is basically uniform, Did the family history still matter?
It absolutely did. This was the first major validation. They found that cells belonging to the same clone had gene expression profiles that were significantly more similar to each other than to random cells, or even to a neighboring cell from a different clone.
So even without a complex environment, the inherited patterns were still visible.
It proved the system could capture these heritable intrinsic expression states. The family history leaves a real, measurable mark.
Okay, before we move from the lab bench into the tumor, let's just do a quick recap here.
That was good.
So, SpaceBar uses 96 custom-built synthetic barcodes that are designed to be seen by standard imaging tools.
Right. And it gets its power to track thousands of clones from combinatorial labeling, giving each cell a unique mix of those barcodes.
And the initial tests confirm that, yes, clonal sisters really do share more similar gene expression patterns. Lineage matter.
That's the foundation. Now for the real test.
Now we take that family history and see how it holds up when things get messy, when the environment is complex and chaotic inside a living tumor. This is where it gets really interesting.
This was the crucial step, to prove it could work in a biologically relevant setting. And first, they wanted to see if it could capture known heritable states.
Things we already know are passed down from cell to cell.
Exactly. They looked at two genes, OASL and IFIT2, which are known markers for a state in melanoma cells that's primed for resistance to therapy.
So a cell with these genes turned on is basically getting ready to fight off cancer drugs.
Precisely. And it's a state that's known to be heritable. And sure enough, they found that cells from the same clone were much more likely to share the same expression state for these markers. It proved SpaceBar could track these important heritable patterns even if they're only switched on for a short time.
So with that validation in hand, they moved to the full tumor model.
They did. They took these barcoded melanoma cells, injected them into mice, and let tumors grow for about 5 weeks. Then they harvested the tumors, sliced them up, and put them under the microscope.
And the scale here sounds pretty huge.
It was. Across just 5 tumor sections, they profiled over 300,000 cells, and from that, they were able to assign about 23,000 unique clones. It's a massive data set.
Okay, so now we have it all. For hundreds of thousands of cells, we know who their family is and we know their exact address in the tumor. All the pieces are on the board.
Right.
But how do you actually measure it? How do you put a number on whether a gene's behavior is driven by family history or by the neighborhood?
That was the next big challenge. And it required developing a new computational tool. They created 2 metrics, the clone score and the space score.
The computational heart of the study.
It really is. It's how they quantify the intrinsic versus the extrinsic drivers of gene expression.
So can you walk us through how these scores work, maybe without getting too deep into the math?
Absolutely. The logic for both is actually pretty simple. You compare the real biological signal you see in your data against what you'd see just by random chance.
Okay.
So for the clone score, you first measure the actual variation in a gene's expression across all the real clones you've identified. Then, as a control, you computationally scramble all the clone IDs. You just randomly reassign the family tags to all the cells and measure the variation again.
I get it. So you're comparing the real family structure to a completely shuffled random one.
Exactly. The score is just the ratio of the real variation to the scrambled variation. If your clone score is much bigger than 1, it means your family identity is a real significant driver for that gene.
So it's basically a signal-to-noise ratio. Is my family's influence a real signal, or is it just random noise?
That's a perfect way to put it. And the space score works the exact same way. You just scramble the cells' spatial locations instead of their clone IDs. A high space score means your location, your neighborhood, is what's really driving that gene's expression.
This is the moment of truth then. When they ran this analysis on those melanoma tumors, what did they find? What was the balance between nature and nurture?
The result was incredibly clear. And maybe a little surprising. The tumor microenvironment was the dominant force.
By how much?
Overwhelmingly so. Out of all the genes they analyzed, 46 of them, that's 40%, had high spatial scores. Their expression was dictated by where the cell was sitting in the tumor.
Okay, 40%. And what about the clone scores? The family history?
In stark contrast, only 5 genes, just 4.4%, showed high clone scores.
Wow, wait, only 4.4%. That's a massive difference. Was that expected? I mean, does that mean ancestry is almost irrelevant once a cell is in a tumor?
Well, it tells us something really important. For this type of tumor, which started from a lab cell line, the immediate environment exerts this incredibly powerful uniform pressure. The location is telling almost all the cells what to do right now.
But that 4.4% must be important.
It's arguably the most important because that's where the long-term heritable differences are hiding. That's the stuff that leads to drug resistance and metastasis down the line. It's the cell's hidden potential.
Let's get into some specific examples. Give us the poster child for a gene that was totally driven by its location.
The two best examples were the genes with the highest space scores, MITF and VEGFA. And they showed this beautiful division of labor based on their position. MITF, which is a key melanoma growth factor, was expressed highest right at the very edge of the tumor.
Suggesting it's involved in, interacting with the healthy tissue around?
But it's the idea. It's on the front lines. But VEGFA, its partner in this story, showed the complete opposite pattern.
And what does VEGFA do?
It's a gene that promotes blood vessel growth, and its expression was 6 times higher deep in the interior of the tumor.
Which makes perfect biological sense.
Perfect sense. The core of a big tumor is often dying. It's starved for oxygen and nutrients. So the cells there are screaming out VEGFA to try and recruit new blood vessels to survive. The space score captured that immediate, desperate environmental response perfectly.
Okay, so that's the neighborhood. Now, what about that tiny 4.4%, the clonal outliers? What was the strongest heritable gene, the one that ignored its neighbors?
That gene was SFRP1. It had the highest clone score in the whole analysis, which means its expression is strongly controlled by some kind of heritable, long-lasting cellular memory. It doesn't care what the cells next to it are doing.
And how could they be so sure that its expression was truly clonal and not just some tiny little micro neighborhood that they couldn't see?
This is the crucial aha moment of the whole paper. It's where the single-cell resolution really proves its worth. They focused in on one specific family, clone 31, which had by far the highest expression of SFRP1.
And were all the cells from clone 31 clumped together?
No, and this is the key. They were completely interspersed. They were scattered throughout the tissue, mixed in among cells from dozens of other clones.
So if they're all scattered, how does that prove it's clonal?
Because without the barcode, you'd never know they were related. The researchers demonstrated this really cleverly. They simulated what the data would look like if they had used a lower resolution platform, sort of blurring their vision.
Like what many commercial systems provide.
Exactly. And when they did that, the distinct clonal pattern of SFRP1 completely vanished. It just looked like a random spatial blob. It even started to look like it was correlated with 8 other nearby clones, which was totally wrong. Only with SpaceBar single-cell precision could they say, definitively, this is an inherited family trait.
And what's so important about SFRP1 specifically?
It's a modulator of a pathway called Wnt signaling, which is tied to things like stable epigenetic memory and, importantly, melanoma dormancy.
So finding a clone that has this gene permanently switched on suggests it might have a pre-programmed ability to go dormant, survive therapy, and then cause a relapse later.
That's the implication. It's a window into a cell's future destiny, not just its current job. And that's what SpaceBar is built for, to find that longer-lived expression memory that really governs what a cell will become.
That really is the core achievement here, isn't it? It's giving researchers the tool to finally properly separate that nature versus nurture debate at a single cell level inside a real tissue.
Exactly. And while we see the environment is the dominant immediate driver, being able to pinpoint even a few of these powerfully heritable genes like SFRP1 is what's critical for understanding the long-term threats like resistance, dormancy, and recurrence.
Which really leaves us and you with the ultimate question to think about. If a cell's location dictates 40% of its current visible behavior, what hidden heritable differences, carried by just that tiny 4% of genes, are secretly setting the stage for that cell's long-term destiny? Thank you so much for sharing these sources with us. We hope this deep dive helps you see the cellular world and the future of biological analysis with a little more clarity.